Do you know that 50% of the USA adults are obese? This huge percentage is mainly caused by the Western diet, like the cookies in this picture, that is rich in fat and simple sugar, accompanied by low physical activity. In my studies and my experiment, I ask why we are vulnerable to such. Why can't we eat whatever we want without being affected? For this, I'm testing the thrifty genotype hypothesis, stating that in ancient humans, there were natural selection favoring high fat storage when food was available to metabolize the fat and endure the time of starvation. Nowadays, however, our body stores a lot of fat, but we always have food with lots of physical activity. The fat is not metabolized, leading to obesity. This is true. Its logical counterpart, the Spencer genotype hypothesis, should be also true, where selection for high performance should acquire the ability to reduce the adverse effect of the excess food, so no more obesity and diabetes. Both hypotheses were studied in selection experiment on bank voles, where the bank voles were selected into aerobic with significantly higher aerobic metabolism during swimming, always represented in blue, compared, of course, to a control randomly bred animal represented in black. Voles are also selected into predatory lines. They can catch the prey within seconds, significantly less time is taken compared to the control line, predatory always in red. And the third selected trait is the herbivorous. They lose less weight when given low quality diet compared to the control group. The aerobic and predatory are more active, thus they have high performance, so they could represent the Spencer genotype, whereas the herbivorous, they have significantly higher fat reserves, so they could represent the thrifty genotype. We expect that the adverse effect of the diet to be less severe in the aerobic and predatory and more severe in the herbivorous compared to the control lines. For this, we took the animals, we gave them either standard, always anti-symbol, or Western diet filled symbol. We measured their body weight, glucose concentration, fat content. We measured basal metabolic rate, here sprint speed. We do the crossover design at this point to check both the immediate and the long-term effect of the Western diet. At the end, we dissected the voles, we weighed their organs, and we took blood for to check the function of the liver. But we were surprised and shocked by the results, and let's zoom in a little bit. So the body mass, glucose concentration, fat content, performance measurements based on metabolic rate were not affected by the Western diet. Let's take body mass as example, comparing the standard diet, the anti-symbol, to the Western diet, the filled symbol. They have approximately the same mass in all the selection. The only thing that is affected was the liver mass and function. Let's consider the mass of the liver. It is significantly higher in the Western diet compared to the standard diet. The conclusion, the bank voles are highly resistant to the adverse effect of the diet. Most of the parameters were not affected by the Western diet, only voles showed signs of liver disease. And in these parameters, all the lines were affected similarly, so no support for the thrifty or the Spencer genotype hypothesis.